Good morning, everyone, and thank you all for attending today's Journal Club webinar sponsored by the 80th Assembly on Respiratory Structure and Function Early Career Professionals Working Group. My name is Niyati Burkar, and I will be moderating today's webinar titled Asthma Under the Microscope, a study on the single-cell spatial view of the airway wall. Today's webinar consists of a presentation by Dr. Régis Joulia, who is a Wellcome Trust Fund Group Leader and British Heart Foundation Junior Research Fellow at the National Heart and Lung Institute at the Imperial College in London. His work focuses on the cellular and molecular mechanisms driving airway inflammation and remodeling, with a particular emphasis on how spatially organized cell to cell interactions contribute to asthma pathogenesis. Dr. Julia is the first author of the landmark Nature Immunology study titled A Single Cell Spatial Chart of the Airway Wall, reveals pro-inflammatory cellular ecosystems and their interactions in health and asthma, which applied advanced single-cell spatial transcriptomics to map the human airway wall. We are so excited to learn from you today, Rehis. A special introduction of our expert panelists who have taken the time to join us today. We have with us Dr. Scott Randell, who is a professor in the Department of Cell Biology and Physiology at the University of North Carolina at Chapel Hill and a principal investigator in the Marsico Lung Institute. He is widely recognized for his expertise in airway epithelial cell biology, particularly in the culture, differentiation, and regenerative capacity of the human airway epithelial cells that have advanced understanding of diseases such as asthma, COPD, and cystic fibrosis. Dr. Randall was also recently featured in Clarivate's 2024 highly cited researchers list for publishing the most influential scientific papers in the past decade. Congratulations, Dr. Randall, and thank you for joining us today. We have Dr. Josalyn Elcho, who is an associate professor of medicine in the Division of Pulmonary Critical Care at the University of Iowa. She also serves as the vice chair for professional development, as well as a director for the Iowa Inflammation Program and co-director for the Center for Immunology and Immune-Based Diseases. Dr. Cho's research focuses on understanding the immunologic mechanisms of pulmonary diseases using translational studies and mechanisms to investigate innate and adaptive immune responses during viral infection and asthma. In addition to her research, she is also an active pulmonologist and educator, contributing to both clinical care and translational research at the intersection of immunology and pulmonary medicine. Thank you, Dr. Cho, for joining us today as well. Next, we have Dr. Praveen Akuthota, who is an associate professor of medicine at the University of California, San Diego, and is a physician scientist specializing in pulmonary and critical care medicine. His research focuses on eosinophilic inflammation and the immunopathogenesis of asthma and related airway diseases. Dr. Akuthota is recognized for his work on eosinophil biology, type 2 immune responses, and the development of targeted biologic therapies for severe asthma. Thank you, Dr. Akuthota, for joining us today as well. Just a note, the session today will be recorded and posted on the ATS website, and the recording link will be sent to all the registrants of this webinar. We will also have time for question and answers from the audience after discussion between our expert panelists and the presenter. And at any point, and at the end of the webinar, please use the Zoom raise hand feature to enter to ask for questions, and we would unmute you. With that, I will let Rahis take the stage away with his presentation. Good luck.
Thank you for the very nice introduction and the invitation to speak today about some of our recent studies and what we have done with asthma, human samples, and single-cell spatial transcriptomics. So my lab is located at Imperial College in London within the NHLI, as mentioned in the introduction. And our lab and department overall is quite interested into this area of the lungs, which I'm sure you are all familiar with, which is this lung bronchial space, which is part of the lower airway. And this is really the crossroad of both immune and stromal cells. So we are an immunology lab, so we are really interested into this space because locally there are all type of immune cells that are present, but also very large epithelial structure, but also very enriched abundant vasculature, which is also something that my lab is very interested about. And this structure is not only interesting on the biological point of view, but also it is a starting point of very prevalent disease such as asthma, and we believe also as well COPD. And I don't have to remind you of this number where we know now that asthma is impacting millions of people worldwide. This is a very prevalent disease with no cure. And this is also very sad to know that about now 10% of the children, at least in the United Kingdom, are impacted by a form of asthma that can have detrimental impact later in life. I'm not going to spend too much time explaining the origin of the disease, but we still don't really understand. But we know that critical feature of the disease are dysregulated immune response, and this tissue remodeling, which is a very big, large umbrella, which basically a bit recapitulate a lot of structural changes that's happening in the lungs that are leading to poor lung function. And just a bit recently, what we have done with the lab, it was really good exercise to a bit trying to do this, is to generate a scientific animation, trying to better explain what are the current challenges in asthma and what are basically the limitation of some of the therapy. And I hope this is going to work. So I hope that now that should come up also as well with the sound.
With every breath, our lungs are exposed to thousands of potential threats, allergens, viruses, pollution, and more. These airborne particles travel deep through the respiratory tract, eventually reaching the bronchial wall of the lungs, where airways and blood vessels meet in a delicate balance. At the surface lies the lung epithelium, a protective barrier that separates the air we breathe from the internal tissues. Just beneath are resident immune cells constantly on alert and ready to respond. When the lung epithelium is damaged, it releases distress signals known as alarmins and chemokines. These signals activate the local immune cells and kickstart the body's inflammatory response. Immune molecules begin to flood the area. Dendritic cells move to nearby lymph nodes to initiate the adaptive immune response. At the same time, blood vessels become activated, allowing circulating immune cells, neutrophils, eosinophils, lymphocytes, and basophils to enter the lung tissue. These immune cells work to eliminate the threat, but in asthma, this response becomes chronic, and inflammatory cells are continuously recruited to the bronchial wall. Constant immune activation leads to structural changes in the lung tissue, known as tissue remodeling. The extracellular matrix thickens, airways and blood vessels shift, and mucus is overproduced, all of which contribute to breathing difficulties experienced by people living with asthma. Current treatments, like biologic therapies, target some of these immune cells, particularly eosinophils, by blocking the signals that keep them alive. While effective at reducing inflammation, these therapies don't fully address the deeper issue, changes to the lung's architecture. To develop truly curative treatments, we need to better understand how different cells are organized and interact within the lung. Only then can we begin to repair the damage caused by chronic inflammation and offer lasting relief to people living with asthma.
And this is something that is like, I'm sure, quite a simplified view of what you know about asthma, but it's actually really good to actually point out some of the issues that we have now, which are mainly this misunderstanding on why and how the tissue architecture is changing during asthma and what are the molecular regulator important in this process. So we are working in the lab using a lot of imaging tools, and this is something that we think is important and critical to understand tissue architecture. And we use both combination of mouse model of allergic airway disease, but also human samples. And we use, in mouse model, we use clearing approaches and precision cut lung slices to better understand the tissue architecture and where cells are actually positioned. And we can do this very nicely in mouse. We have a lot of samples. We can do this sort of really like sort of whole organ imaging where we can visualize the vasculature, but you can see it in green here, the airways in purple, and one type of immune cell called mast cell here in blue. And we can better define where the cells are locating, in which association they are with structural, but also all the immune cells, and where they are present across all the lung lobes. How to do this in human is a very big challenge because obviously these patients are all under formal treatment. There are different stages of the disease. The samples that we can get, which is a very, in comparison to cancer field, are very, very small. And we try to get as much information as possible from this sort of very, very small sample and trying to understand better how these cells interact with all of them in this space. So to do this, what we started to do, it's already quite a few years now, we started to explore if spatial transcriptomics was a good tool that could be useful to better understand asthma in the lung bronchial wall. So we use a platform called Xenium, which is part of the 10x companies. And this Xenium platform relies on the use of barcode. This very simply, these gene-specific barcode are going to hybridize within tissue sections. And using this padlock system and following after ligation and amplification, you can now detect which mRNA is present within tissue section and after associate them, as I'm going to show you, with the actual cell. So the sample that we employed during these studies were a collection of endobronchial biopsies performed by our excellent team of clinicians here at Imperial College, using both healthy donor and severe asthma donor. All of these severe asthma were under a form of treatment, which is normal practice, where they are both under inhaled steroid and some of them under biologics. What in addition we performed during this study, I'm going to briefly mention this, is that we had a really excellent collaboration with the lab of Professor Josh Boyce and Elliot Israel in Harvard, where they gave us samples from previous clinical trial performed more than 10 years ago, where they treated patients with imatinib for 6 months and collected samples pre and post-imatinib samples. We got samples at the end of the clinical trial, and we can reuse after these samples to analyze if imatinib had an impact on the spatial architecture of the tissue. So this is what the Xenium slide look like overall. So we spent a lot of time trying to optimize the placement of these small biopsies on each Xenium slide to maximize the number of samples that we could use, because this is very expensive technology. And you are going to have just a quick overview of what the sample looks like following the process of Xenium. So you have here an image of a tissue section stained with DAPI that we use for cell segmentation. So you're going to see all the white dots are here, each cell that we could identify. And we use this cell segmentation, which was at the time the only thing available for us. So we use a DAPI-based expansion to detect where are cells within the tissue. And we can now detect, so each little dot here is a transcript that we can associate with the specific cells that has been segmented. And we can then analyze which cells is expressing which transcript, but also as well spatially where they are located. And the first step of this process, following cell segmentation and transcript association, is to determine which cell type can be correctly identified. And what we did first identified was about 18 different cell clusters, which are not the most cardinal one that you will find in human samples, human bronchial samples. So you can find ciliated cells, basal cell. We find a very nice population of endothelial cells, smooth muscle cell, fibroblasts, etc. So important things to point out at this point is that we didn't identify all the immune cells that we were hoping to see. And this is, we believe, due to the fact that our cell segmentation was not allowing a very fine definition of lymphocytes. And we believe that most of our lymphocytes are actually residing within this cluster 12, as we call it, which was basically a mix of T cell and B cell all together, but we couldn't really formally identify them. We make sure the identity of the cell type was correct using the cell lung cell atlas data, which is here full transcriptome of a single cell, and we found that they were matching perfectly the signature between this cell type. Something a bit disappointing when we actually compared healthy and asthma samples, that we didn't identify major cell differences between this data set, except for a few different cell types that I don't think I'm going to have time to mention today, which are these serous cells here present there in this corner, which were highly different in asthma because of their very high production of mucus and lactoferrin expressing genes. Something that we also as well quite happy about is that the samples that we got, this endobronchial biopsy, were really good in terms of signal for some mediators such as chemokines. proliferation genes. We found also very nice signal for extracellular matrix genes, but also immune regulation genes that were also present. And at this point, what was important for us, it was not to only consider this data set as another single-cell RNA sequencing data, which has already been done for asthma, but also develop the tools to analyze the spatial interaction between these different cell clusters, where you can see here that now we basically also use this tool to you develop tools to analyze spatial interaction between basal cell and endothelial cell using here distance, but we also made huge progress since this publication to also expand this tool toward also different type of spatial interaction, which is now possible to do this using this sort of technology. The thing we also when we started to analyze this type of samples and what we better define our analysis is that we observe that within all or most of our actually biopsies, we have two very different transcriptionally regions. So we had a very nicely defined epithelium, which we defined as the epithelium niche, which was a high level of transcript here you can see within the epithelial niche. Then there is this sort of kind of like desert of transcript where nothing too much was happening, and this was actually associated with this sort of smooth muscle layer where we couldn't identify here a lot of transcripts, a lot of transcriptional activity, but there was this sort of huge barrier of cells that was quite different. And then a bit deeper within the biopsy, we had this sort of very highly, and here you can observe this is an asthma phenotype, an asthma biopsy, where we have this very rich and dense mucosal gland structures that have a very different type of actually transcript present there, and the cells were actually also different. So we made very careful the distinction between these two regions and not trying to actually mix the two different cells between these two niches because they are actually quite different. And the first question we ask within each of these niche, where are chemokines and alarmin produced? And this was really important because both chemokines and alarmin, such as IL-33 and TSLP, are very big driver of asthma. They are really good biologics now that have been designed to target this mediator, but we wanted to define where they actually produce, which was still something that is not super well described. And when we looked into all of these transcripts, so here this is like regard of what type of chemokine or what type of alarmin, we find this sort of region of enrichment of this cluster. So when you look at the total number of transcripts, for example, here, we observe that not the whole epithelium was lighting up, but there was some kind of area where we had most of the transcript expressed. So you can see, for example, here there is a lot of transcript for chemokine and alarmin. Then it starts to slowly go down. There is nothing about in this region, for example. Then again, a very high level of production. Then again, all of this area is completely almost empty in terms of chemokine. And then after it was a bit similar all across the epithelium. And that was true for healthy samples, and in asthma it was also a similar type of structure. So we had also as well what we call this inflammatory hub, where we had this sort of enrichment of cells expressing chemokine and alarmin. The main difference here between these two different type of donor, but the inflammatory hub in asthma were actually the cells were more aggregating together. So we had actually an enrichment of mediator expression due to this close association of cells together. So what are actually these cell types? So we assume at the beginning it was going to be immune cell mediated alarmin and chemokine production, but it was not the case. Immune cells were actually quite poor in comparison to stromal cells, epithelial and endothelial cells to produce chemokine and alarmin. And we identified here on this graph that you are maybe familiar with, where here you have the chemokine and alarmin on the left side, and the size of the dot corresponds to the frequency of cell expressing the mediator, and the color of the dot corresponds to how highly the mediator is expressed. So if you look at the big yellow dots, you will find that basal cell 1 and endothelial cell 2 were the most potent to secrete IL-33 and also expressing a lot of chemokines such as CXCL6 and CXCL12 or CCL19. Other cell types were able to produce chemokine, but to a lesser extent. Ciliated cells, for example, were not able to produce as much. Goblet cell had very high production of CXCL6, which was interesting for us because it's a neutrophil chemoattractant. But we found that these two populations of endothelial cell 2 and basal cell 1 were really the one driving most of IL-33 production. something that we had to put in the paper, but we still are not sure exactly of how to interpret this data, is that in all our asthma samples, which are every other column, you will see that the size of the dot and the color of the dots indicate a lower amount of production. And we are not sure at this point if it's due to the actual treatment of all these patients are actually a high dose of inhaled steroid, or this is due also something to the disease. And this is quite hard to at this point to discriminate between these two elements. And we are trying out using this also as a using mouse model to better understand this, but this is still something that we still haven't fully understood. So more good things about this sort of now spatial data is that we can identify which cells are expressing which mediator, but also as well how they are organized in the tissue section. And we did this, so we looked back into both the HNE and the Xenium data, and we found that actually basal cell 1 and basal cell 2, or endocellular cell 1 and endocell 2, are not existing into separate regions. They are existing always as a mosaic pattern. So you have always, for example, a few endothelial cell 1 within one blood vessels, then a few endothelial cell 2, then again a few endothelial cell 1. It was the same for basal cell, where you have this sort of high basal cell 1 expressing IL-33, then a few basal cell 2 that are not expressing IL-33. So it was always this sort of mosaic expression of cells, which are maybe an indication of basically sub-region within each niche, which are again confirming this sort of like inflammatory hub basically concept. The big difference that you can nicely see on these two images is that in asthma, these cells producing chemokine and alarmin are getting closer together, which was really for us completely surprising to see blood vessels getting closer actually to the lung epithelium and creating this sort of basically enriched inflammatory hub where all these cells are actually clustering all together. And this was both true at the lung epithelium, but also when we looked deeper inside the mucosal gland, we had actually a similar process between endothelial cell 2 and mucosal gland cells, where they also get closer together and form this sort of big cellular aggregates. And we focus a bit more here on endothelial cell just for this presentation, but the endothelial cell 2 were actually very quite different from endothelial cell 1. They both express cardinal genes for endothelial cells, such as von Willebrand factor or PLVAP, which is a transcriptional regulator of endothelial cell, or CD34. Both of these cell types express them, so they are both endothelial cell. But the endothelial cell 2 express very high level of this cardinal endothelial cell gene compared to endothelial cell 1. For some reason that we are still not sure why. What was interesting for us at this point also as well was to show that they also express ACKR1. If you're not familiar with this receptor, it's an atypical chemokine receptor that we have done works previously before. It is a known post-capillary venule marker, but it's also a very important non-signaling chemokine receptor, which is able to present chemokine locally to immune cell and retain them. So we think this is maybe also as well a key molecular regulator of why this endothelial cell could be more pro-inflammatory, is because they have both the ability to produce a lot of chemokine, but also retain them on their surface to attract lymphocytes. We then drew this complicated graph, which took us actually months to actually find and decipher how the best way to represent spatial interaction. And we ended up doing this. We already improved them since then, but the goal here was really to see from each endothelial cell 2 what are their closest cellular partner. And you have here basically the distribution of this neighborhood, of this neighbor per the distance. So if you look at close distance, which is less than 25 micrometer, you have this sort of repartition of cells. So a lot of endothelial cells are also associated with themselves, which are these two ear segments, but they're also associated with basal cell to a little extent, then mast cell and macrophages also, and not really a lot of smooth muscle cell. What you can see is a difference here between healthy and asthma is that these endothelial cell 2 are actually less associated with themselves, but there is this sort of increased interaction here with basal cell. Even if it seems quite low on this graph, you can imagine that this sort of interaction where you go from almost maybe like 5% of endothelial cell 2 in close proximity to basal cell become 10 to 15%, and that's going to make a massive difference when you think about production of inflammatory mediator. And these cells, and that was a question of one of the review, are they actually proliferating or are they actually changing spatial coordinates? And they're actually changing spatial coordinate. We didn't find a lot of proliferation happening, but we did find that the shorter cell distance was decreasing. So you have here the shorter cell distance between each endothelial cell 2 and basal cell or each different cluster, and there was a reduction of this distance. So it means that every endothelial cell 2 are actually getting closer to the other cell cluster or this indicated cell cluster, especially the epithelium, confirming some of our imaging data showing that there was indeed change in terms of structure of the bronchial wall. So how could this work, in which basically cell could regulate the formation of this inflammatory ecosystem? And we hypothesized that mast cell could play a key role in this process because we worked on this cell before. They express all the range of mediators that are important for tissue remodeling. And indeed, when we looked at where are these mast cells located in our spatial data, we found that they are always in close proximity to either basal cell 1 or endothelial cell 2, both in healthy and in asthma samples. They express different genes that are important for tissue remodeling, notably carboxypeptidase A3, or we found also as well amphiregulin, that was also nicely expressed in both mast cells from healthy and asthma samples. And they also as well express mediators that are known for mast cells, such as the ST2 receptors, the L33 receptor, tryptase, and also this carboxypeptidase A3. The mast cell association were really also there when you look at their spatial interaction, they are present all across the tissue section, but we find them nicely associated with the two types of endothelial cell, but also basal cell, and the very also interaction with macrophages and fibroblasts. And this set of interaction was also changing with asthma. We had a very different set of interaction. They were getting closer to the endothelial cell, a bit more distant from the basal cell. So we think that they're also always in close association, but this sort of set of interaction was changing. And so to go back after to mast cell, we thought, well, could we analyze actually some kind of a study that looked into mast cell inhibition? And this is what we got in contact with Josh and Elliot Israel at Harvard, and also as well Katie Cahill, but she's now working at Vanderbilt, where they did this trial about 10 years ago, where we treated a patient with imatinib for 6 months. And at the end of this trial, we observed that this patient had a decreased airway hyperresponsiveness, a decreased mast cell count, but also has a reduced tryptase release in the serum. So we got samples at the end of this trial, which was a 6 months time point, and managed to collect and transfer this section from the original tissue slide to the Xenium slide and analyze them using our platform. We managed to get nice actually mRNA information, despite the fact that these samples were actually quite old and preserved, not in optimal condition. So we identified in this data set 21 different cell clusters. So we had even more cell clusters comparing to all initial samples. And we had actually an enrichment into immune cell, which was surprising for us. We are not sure at this point if it was due to this patient at the time were under maybe different form of treatment. That's why we actually had better resolution for some immune cells, such as CD8 T cell and plasma cell. There were big differences between placebo and imatinib-treated patients. So the CD8 notably population, plasma cell, and fibroblasts, which are all targeted by imatinib, are actually quite different between these two different between the different group of patients. When we looked into these samples, we also found a similar type of inflammatory hub, where basal cell and endothelial cell were the most able to produce chemokine and alarmin, and they were again very much associated with themselves at the epithelial wall. And imatinib, as you can see on these images, and the number of actually dots was massively reducing the number of transcript, notably chemokine and alarmin in these cells. Endothelial cell 2, for example, were almost not expressing L33 anymore, which was really interesting. We looked, yes, so that's the actual quantitative data showing here L33, for example, production was reduced in basal cell and almost completely abolished here in endothelial cell 2, where you have here the imatinib treated colon here, which is in this middle. So it indicates overall that imatinib had a really drastic impact on both basal cell and endothelial cell capacity to produce chemokine and alarmin. We also look at the mast cell profile in these samples, and we found that mast cell, comparing to the placebo control group, so imatinib treated patient had a reduced mast cell activity, characterized by reduced carboxypeptidase A3 expression, but also reduced IgE expression, but also ST2 also as well was going down, and PTGS1, which is also enzyme producing COX mediator such as prostaglandin E2 was also reduced in imatinib patients. Their set of interaction was also very different. So we found that during following imatinib treatment, we had actually a reduction in terms of cellular interaction. So this is quite, again, very complex graph to follow. But overall message is that imatinib was able to restore some kind of a more healthy type of mast cell interaction, where they had actually less interaction with endothelial cell and basal cell, and we found them get a bit more into a normal type of placement within the epithelial wall. And finally, so just to finish this on these studies, what we try to do is to also offer to the scientific community a way to basically extract more meaningful information from this data set. And we applied a tool developed now a couple of years ago by the team of Sarah Teichmann, where they actually developed this sort of pipeline where they could link single-cell RNA sequencing information with drug targets. So they can link actually two databases together and extract for each cell cluster what are the most likely targets, the most drugs that are most likely to target them. And we repurpose this tool for spatial interaction, where we could now extract for each individual cluster that we identified within our data set, what are the most likely drugs that are able to target them. And here, if we looked a bit more in zoom in of this dot plot, we can see, for example, that the endothelial cell 1 and 2, we had a very high level of interaction with this drug called caplacizumab, which is actually an anti-VWF drug. We are not suggesting here that these patients will start to get some anti-VWF treatment, which is only used for coagulation, but we just here trying to show the potential of what this sort of technology could achieve in a more personalized medicine type of drugs. And we also extracted spatial information from this drug. So basically where this drug, and here you have an example for this recently approved drug for cervical cancer, which is called tisotumab vedotin, where this drug is the most likely to interact within the tissue samples and also which cells are going to be around the target of these drugs. And we think this is quite important. It could have some nice potential future therapeutic opportunities there. So in summary, we think that we identified better where alarmin and chemokine are produced at the lung bronchial wall, but we identified this sort of inflammatory ecosystem or inflammatory hubs that could be really important also as well in healthy condition to drive some immune training and maybe immune development and maybe immune tolerance as well. And we think that the chronicity of asthma is inducing this sort of dysregulated inflammatory hub with a higher production of inflammatory mediator. We think that IL-33 and chemokines are key in this process, and the combination of stromal cells, so endothelial cell and basal cell, are driving this sort of like aberrant tissue architecture response. Imatinib was a very interesting treatment that had very wide impact. This was really interesting for us, but also as well shows the potential to reinvestigate some old clinical trial samples to see if we can extract some more outcomes and more information from this sort of drug treatment. And we believe that drug-to-cell was also show great potential to understand drug interaction within the tissue. This is quite important in the context of personalized medicine. And on this, I would like to thank my lab. So that was work done a bit more by some of the lab members. And we work closely with the group of Professor Claire Lloyd at Imperial and a really good team of collaborative clinicians. So Sejal Saglani, Lola Lowenthal, which is our adult ear clinician that gave us some biopsies that we worked on. And we had this amazing collaboration with Katie Cahill, Josh, and Elliot at Harvard Medical School. And we'd like to thank our founder, notably the NIHR, which funded these studies. And my lab is funded by both the Wellcome and British Heart Foundation. And on this, thank you very much for your attention, and I'm happy to take any questions.
Thank you, Régis, for the excellent presentation. And with this, I'll open the floor and for the conversation amongst the expert panelists for any feedback that you have, any questions that you may have for Régis. As a reminder, for the audience members, if you have any questions, please use the raise hand feature displayed on your computer screen, and we will unmute you for any questions that you have. Thank you.
So Régis, I can start.
Yep.
Brilliant work. I've kind of seen some of the stuff in progress at meetings, and to see some of it come to fruition is really incredible. So it's fantastic work. I have a lot of thoughts that are a little scattered, but let let me maybe start and see what other people think about this. I feel like we have a lot of conversation these days about dysregulated epithelium in asthma. This work clearly adds to those concepts, but to overlay the heterogeneity, the niche part of it on top of that, I think is what is a real leap forward, and particularly the mast cell. I think we've forgotten about the mast cell a little bit, and the mast cell epithelium interaction being so critical in what you're seeing. Obviously, you're losing some cellular players here by nature of the technique, but that almost gives you a little bit of focus to kind of understand some very specific interactions.
Yeah, no, I fully agree. I think yeah, this sort of technology, and we put it out there, and we have been very honest about the limitation of was happening. I think technology is moving very, very fast. Some of the recent data are shown, and we generated generate. I mean, like I think the lymphocytes I think are going to be resolved quite soon. We now managed to get neutrophil and eosinophil, so we are quite happy with them also there. So it seems to be a lot of things happening there. But inherently, a lot of these cells are also they're not very transcriptionally active. So we have to be also careful and mindful of if a cell is not transcriptionally active, we are not going to pick it up. And that's why I think a lot of still proteomic tool and multiplex imaging and still some maybe old school techniques are still essential to better understand some of this pathology.
So I'll just echo what Praveen said. I think the mast cell stuff is really interesting and important. So in our own work, we've done kind of a slightly different comparison where we've looked at allergic individuals and divided them into two groups, those with asthma, so those that have tissue remodeling and airway hyperresponsiveness, and those that get type 2 inflammation, but don't have the cardinal features of asthma. And mast cells were, I think, the single strongest distinguishing feature to those between those two groups before allergen challenge. And so I do think they're really important. And I was just wondering if you could expand on kind of what you think they're doing within the tissue to mediate this increased association between endothelial cells and epithelial cells. Is it that the tissue in that space is fibrosing and kind of pulling the two cell types closer together? Is it actually doing something different that you think makes them more tightly associated?
And yeah, it's a really good point. I think, yeah, mast cell has been massively understudied in a lot of this pathology, and that comes from, I think, some technological tools that were not available to just study them properly. Using this sort of new tool for spatial proteomics, transcriptomics, they are going to appear in all data sets because they are really well abundant and they are super present in all of this tissue that we looked at. To go back to your question about how they could do this, we really don't know. So we have hypotheses, but how to test them is just really a big challenge because they are very resistant to any kind of genetic manipulation. The mouse model that we have are good to some extent, but they are not really showing the number of mast cells that we had in human comparison. I mean, we have about 1,000 times less mast cell in the mouse model compared to a human, so it's very difficult to just see what is doing what. Our main hypothesis so far is we think that they can regulate the availability of chemokine locally. So we think that some of these aspects are actually important for driving some cell movement where they can actually regulate extracellular matrix deposition and placement of chemokine to actually prevent or promote cells to actually migrate in some area. Or that's our main hypothesis so far, but it's just really hard to just make sure how mechanistically to prove that is the case.
I would like to keep going a little bit on the mast cell question, and also relates to two important things. I think the location of the biopsy sites, and the unexpected finding of lower amounts of alarmins and chemokines in. So one thing about the mast cells, another is the Xenium panel has 339 genes. Are both chymase and tryptase on that panel? Because I did have a question about these chymase tryptase versus just tryptase type mast cells. That's kind of one question. The other, probably more important one is these biopsies were all in fairly large bronchi that have submucosal glands. There's some old data from Sally Wenzel that actually the numbers of mast cells increase as you go from bronchi to bronchioles. That's one thing. So it would probably be very interesting to study small airways as well as large. But the question, probably more important, is that the thought was that the lower levels of alarmins and chemokines were a consequence of the anti-inflammatory therapies. So the inhaled corticosteroids are like the mainstay, and are they hitting the larger bronchioles? The deposition of these drugs is a very important issue. And are they getting out to those smaller airways if we were able to sample them? And just thinking about the future, how we can study the small airways with these elegant spatial technologies. A lot of questions at one,
That's
Régis.
okay. So go back to the first question, tryptase and chymase. We did add chymase in the panel. That was, I think, something that we added on because I thought, yeah, as you said, it was important to decipher which type of mast cell there were. We didn't get any signal for chymase. There were only tryptase positive. So it's difficult to know at this point if is it the chymase, did we not pick it up because the signal was not there, or was it not transcriptionally present there? So that's something that we can't be 100% sure. Because mast cell, I mean, do they need to continuously express the mRNA, or if they have enough stock, do they stop expressing it? So that's something that we're not sure about. Then so the smaller airway versus larger airways, yeah, that's something that we are fully aware of it. And we know that we are working towards getting samples from different area and trying to get also as well at this point a bit into smaller airways. We never really looked into detail the number of mast cell across the bronchial type of element, but that's something really interesting. I think that could be really investigated because I'm sure they are very different between these different areas of the lungs. So that's something that we can look at. And drug, that's a really good question about the drugs, because that's a question we're always thinking about in the lab is, are these drugs actually acting where we want, or are they actually stuck somewhere we're actually looking, what are we looking at this point where we are punctioning these biopsies? And that's, I think, we try to look into the opposite way. So when we looked into the drug to cell, for example, we looked at the level of the drug available target, and we assume that if the target was actually reduced, it means that the drug was having an effect, for example, if you see what I mean. So it means that the drug was doing something if the target was less present. And we got some positive information from this saying that, like, yes, the target was less present, so the drugs could have an impact. But that was still quite a correlative information. It's not really like we are not 100% sure. So that's something that I think as a field, like, yeah, we are not 100% like for now having the tools to properly decipher this question. But yeah, that's a really important question.
Also, one quick thing about the proximity of endothelial cells. So in the large airways, presumably these are endothelial cells of the bronchial circulation. And did you see any signal in the basal cells, for example, of VEGF or other kind of chemotactic factors that might have promoted angiogenic type response? So I know probably not the right sample, but were there more blood vessels in the asthmatic than in the non-asthmatic biopsies?
So we didn't find any enrichment in endothelial cells. So the proportion of endothelial cell was similar between healthy and asthma. Saying this, we always be very mindful about this number of cells and proportion because our samples are quite small, so it's difficult to make like a big picture of it. For the source of pro-angiogenic signal, basal cells were not really expressing any kind of pro-angiogenic factor. We found again mast cell and macrophages were the most potent to secrete some of this factor. So we are looking into it now to see if maybe a population of mast cell will start to secrete a bit more angiopoietin 1 or VGFA that could drive some of these changes in endothelial cell. But so far, same thing is quite correlative. So we try to find a bit more now, a better mechanistic approach to confirm some of these questions.
Régis, can I ask you a question? It's kind of about the technique and how good do you think the technique is or not good at detecting some rare cell types in these niches? I'm thinking about Zin Sun's work about neuroendocrine cells and just kind of small cell populations that might maybe seed or promote or be kind of unexpectedly at the center of these niches.
Yeah, the reviewer asked us about neuroendocrine cells, and we tried to also as well add genes that were specifically expressed by PNA. We couldn't detect them, so we are not sure if it's due to either their poor transcriptional activity and means that maybe we cannot see them nicely, or the fact that we maybe don't know enough about the biology of these cells to make sure that we can see them properly using this type of data. I think there is maybe a bit more basic biology to be done on this cell type before going straight for the most expensive techniques to just try to find them where we still don't know what they actually look like and which marker they actually express. Yes, I think the potential is there for this technology to detect this sort of cell type. I mean, for example, serous cell was something that we didn't expect it to find to such high proportion and very nicely defined and very high, but they were there, and we found them quite nice difference in phenotype. So yeah, I think with the correct marker and the correct gene express, you can detect most every cell that you want actually.
I wanted to go back to something that Dr. Randell mentioned in one of his earlier questions, which is the presence and the ability to study the submucosal niche or the mucosal gland niche. And you kind of touched briefly on the fact that that space is very different than the epithelial space. And one of the sort of recent things that caught my attention is that space may serve as kind of a B cell niche. And so I was wondering if you maybe want to elaborate on what's different between those two spaces, if you specifically compared lymphocyte populations from the epithelial to the mucosal gland space, or what you think might be important about what you found differently in those regions.
Yeah. So when we looked into this, if you look into the top genes that are different between asthma and healthy in just overall samples, the mucin genes were actually massively increased in this asthma, and whatever treatment the patients were under, it didn't make any difference. Whereas, like, I think we had even to put it on a different scale on the paper because if not, it was completely dwarfing everything else. And when we looked into where these mucin genes were expressed, it was all completely lighting up this mucosal gland area. And we decided at this point, okay, no, it's best to separate them because they may have massive differences there biologically in terms of what's happening. And when we looked into, and we are currently also looking at this fibroblast and endothelial cell present in this region, they were actually different from the one from the epithelial surface. So if we all put the same fibroblast and endothelial cell within the same family and the same cluster, that was making basically putting differences or actually normalizing some gene expression that if you just do single cell, you are going to put all of them in the same basically cluster, but they were actually expressing different factor and a different set of interaction. So this is why it was really important for us to separate these two different regions. we were also as well looking in the literature. You are right, there was this B cell story with the mucosal gland niche. We found B cell signal within this region. The only issue here that they were mixed with T cell, so it was not possible to make sure that we had really 100% pure B cell population. But yeah, we found very high signal for B cell gene next to this. So we hope that with now better segmentation tool and to reinvestigate some of these questions moving forward. But yeah, I think it's a very super important point that we didn't address yet in this study.
And when you talk about differences in the structural cells there, are you talking about like the growth factors that they produce or chemokines or both?
Yes, it was a combination of antimicrobial peptide. So there was a lot of antimicrobial peptide produced. Lactoferrin was one, but we had a whole different range of like different microbial peptide, which could also suggest that these patients are experiencing some kind of microbial exposure that they can't control, which is interesting. Maybe within the mucus actual produced. And that was so the main thing, yeah, was this antimicrobial peptide, a few chemokines, growth factor also as well. I think there was some weird fibroblast growth factor that we also didn't take a look too much in detail. But yeah, I mean, all of this is out there if people wants to look because we couldn't look at everything.
Yeah, I would like to talk a little more about immune cells. There were differences in the ability to detect immune cells in the imatinib and in your study. Did that reflect a difference in the biopsy procedure or type or location? And the bigger question, I think, is, for these spatial technologies, sampling is really important. If we ever wanted to understand more about BALT or IBALT in asthma, what would we have to ultimately do to be able to, really get a handle on these lymphocyte populations that are so important in asthma?
Yeah, you're 100% right. So the main, even here we did a small scale study looking at what is actually the main differences sometimes between our biopsies, and we found out it was actually the actual clinician taking the biopsies is the main driver for batch effect between our different samples. So even if they tell us they go in the same area of the lungs, it's actually we can literally find that they are not. It's not actually the case. So yes, the main differences between our samples here at Imperial and the samples from Harvard was this enrichment of immune cell. And we think this is due to the fact that, yes, they may have looked into a different area, maybe in the bit lower airway, comparing to what we are doing here. And that was why maybe we find this enrichment and better definition of actually the cell. The immune cell resolution was a lot better comparing in the samples. So
Yes.
that's why I think, yeah, I think it's quite important and critical now moving forward with spatial data that people report very accurately where they took these samples, in which part of the lungs, with a lot of as much detail as possible, because that's going to make big differences.
Yeah, the closet pathologist in me, from looking at sections of whole explanted lungs and fatal asthma, or to get to the lymph nodes enriched portions, they're really in the cruxes of sort of medium to smaller bronchi, and that might be a challenge to ultimately get. Maybe being able to take advantage of explanted fatal asthma lungs. Hopefully they're rare, but they do happen. The very rare larger biopsies. I just put in a shameless plug for the paper we have just released in the Blue Journal. I was really amazed by how many mast cells are in the small airways, as you can see from our spatial proteomics phenocycler in that paper in the Blue Journal.
Yeah.
So we have one question from the audience. Ayu Hutami Sarif, I've unmuted you if you want to
Yes.
ask your question. Yeah.
Thank you for the great talk. I have one question about you showed the pattern of the chemokine expression, especially in the epithelial niche, and then you showed that how actually there is a chemokine gradient expression there. Did you correlate or associate this chemokine expression with the degree of the, for example, immune cell infiltration within this niche, or maybe the more chemokine this niche express or epithelial express, the more tissue remodeling happening, and therefore also immune cell infiltration? Thank you.
Yes, so that's all good question. I would have loved to answer. The issue comes back to we couldn't identify nicely all type of immune cell, so it was just impossible to just make a good analysis there because we didn't identify immune cells, so we couldn't see exactly where they are, especially the migratory one, such as lymphocyte or neutrophil. So it was not possible to do this sort of correlation and see if the inflammatory hub were associated with increased immune cell presence. For the correlation between tissue remodeling and chemokine, I think we're a bit limited here by the size of the biopsy that we had. They are very, very small, so it's quite difficult when you have less than a millimeter of tissue to make sure that like we are actually having some kind of meaningful analysis. I think some of the things that Dr. Randell just mentioned, such as larger biopsies, where you can actually draw a better sort of region of interest, will be a lot more powerful to actually do this sort of correlation analysis than our very small sample.
Okay, thank you.
I just want to comment about the Kia study. And sorry, my Wi-Fi dropped for a second, but
No,
the Kia study is an interesting study, right? In that um, that showed some impact in patients who had lower eosinophil counts and higher, BAL neutrophil counts. And now there's some potentially conflicting data about imatinib and its impact on asthma based on the precise study, which is not yet published, but had some data presented at ATS this past year. So, I think it's a good kind of example how some clinical data can speak to good mechanistic translational work and try to understand um, you know which patients are you having, because not all asthma patients are equal. So the biopsies that you had may not be people who are responders. There might be heterogeneity amongst patients, and try to understand that as a next step would be, I think, really a cool direction.
Mm. Yeah, no, I fully agree. I mean, you were there when Katie approached us at ICANN meeting to give us some samples, so it was really actually a really good outcome of this meeting. And so, yeah, I think the study was really good. I mean, imatinib is doing a million things that we still don't understand, and I mean, just summarizing on mast cell inhibition is quite maybe a shortcut that is quite difficult. So we have to think about also as well the large impact of these drugs can have on a lot of different cell type. But yeah, I think we put it out there. It was really important for us to, as you said, I think a lot of these clinical trials have leftover samples, leftover biopsies that have maybe are really like super, really powerful human experiment that could be explored for us. So I think that's something moving forward that I really like. I think it's really important for the community to share these samples that are just left in drawers and that are actually really powerful experiment.
One quick thing is, and I'm not sure we'll ever be able to do this, but do we need to biopsy folks in exacerbation to see those big changes that you might have expected in this study?
So I guess there's quite a big ethical consideration maybe with the exacerbation. However, moving forward, and something that we now do at Imperial and hoping to do more is to do allergen control studies where people are exposed to allergen or like during asthma and having some kind of a control pre and post allergen exposure in an environment where we know at which stage of their disease they are. And I think that would be really powerful in terms of getting very strong data on limited number of patients, but very well pre and post allergen exposure. I think that's something that is ongoing, not only here, but I think quite different places, and that I think will give us significant insight.
All right, so we have closed in on the hour today. If we don't have any more questions, I would like to thank Régis for this fantastic presentation today. Excellent talk, beautiful pictures, videos, and also for the experts for their valuable time and participation to share their knowledge and make this webinar a very engaging one. Also, thank you to the audience, to all of those who've joined us today for this webinar. Everyone, thank you again for joining us, and please have a great day. Thank you.